Welcome back to my channel Xgene and proteinX. In this tutorial video, I'm going to talk about telomere, aging, and associated factors. In 1930, Barbara McClintock, working with maize, and Hermann Muller, working with fruit fly, both investigators proposed that chromosome ends have special structure required for chromosome stability. Later on, Muller coined the term telomere, and this term came from the Greek word. The telos means the end, and the meros means part. Later on, McClintock noted that without this special end structure or the telomere structure, the chromosome would fuse and often break upon mitosis. So there's the telomere actually is required to protect chromosome end to provide chromosome stability, and this it has been found that the telomere having some special sequence, and that sequence is repeatedly found in the end of the chromosome, and for this sequence it varies different species to species, and this is the human telomere sequence, and this sequence actually repeatedly found in the end of the gene. And usually the length of this telomere sequence is actually from the 0.5 to 15 kV, and also this length of this telomere, it also varies individual to individual. And another important factor is that the length of the telomere is becoming shorter and shorter with age, and the rate of this decreasing or shortening of this telomere is 24.8 to 27.7 base pairs per year. So it associated with some age-related disease or decreased lifespan in humans. So let's talk about the factors that impact on telomere sequence or telomerase activity. So first factor is the obesity that highly influence on telomere sequence. The second is the smoking, and it has been also found that with daily smoking of each pack of cigarettes, an additional 5 base pairs is lost. Another factor is the lack of exercise. Next is the unhealthy food, and the most important factors is the stress. So in the stress, actually it's associated with some glucocorticoid hormone by the adrenal gland, and that leads to the reduce the levels of the antioxidant proteins. So antioxidant proteins is associated with telomerase activity. So it may lead to increased oxidative damage to DNA and accelerated telomere shortening. So let's talk about the associated disease that occurs with reduced telomerase level. So the aplastic anemia, the immune deficiencies, and the pulmonary fibrosis. Let's have a look at the telomere and P53 correlation. So in one study, it has been found that consistently the reduced telomere length in lymphocyte that may impact on hypomethylation of P53 promoter. So hypo or hypermethylation is associated with the epigenetic modification, and that leads to the P53 promoter higher transcription level, and leads to the overexpression of the P53. So as we know that the P53 is the tumor suppressor protein, so as a result, the inhibition of the growth or induction of the apoptosis occurs. So there are some important functions of antioxidant on telomere sequence or telomere activity. So it has been found that the vitamin E, vitamin C, and beta carotene were associated with longer telomeres and the lower risk of the breast cancer. And also the antioxidant, it can potentially protect the telomeric DNA from oxidative damage. So there are another most important factor, that is the exercise. That exercise is highly impact on on telomere sequence, or it reduced the shortening of the telomere sequence. The exercise leads to the reduced telomere shortening, the suppression of the P53 and P21, that apoptotic proteins. So it may result the higher cell growth and the elevated telomerase activity. So it is also associated with the telomere sequence shortening. So elevated telomerase activity, it means like the reduced the telomere shortening. So this is all about this telomere and aging and associated factors. So if you like this video, kindly hit the like button, share it, and if you have any queries, kindly write in the comment section and subscribe my channel. Thanks.